Hello everyone, my name is Priyanka Nagare and my today's topic is Touch DNA Revolutionizing Evidentiary DNA Forensics. Before starting with the topic, imagine a situation where 85% of criminal cases remain unsolved because lack of physical evidence present at the criminal site. As per global crime statistics, millions of cases such as rape, sexual assault or murder remain cold because traditional DNA methods are not capable enough to find actual connecting link. But with the help of touch DNA, it is possible. You might be wondering what touch DNA is, right? So touch DNA are minute traces of body cells, body fluid or epithelial cells that remain on an object when a person touches it. Even 7 to 8 cells are sufficient because you can create proper DNA profile by using those cells. The primary sources of DNA are the murder weapon or shoelace, rope, even plastic or metal objects. The history of DNA forensics began in 1980s, where traditional methods were more focused upon collecting DNA from body fluids such as blood, saliva or semen. In late 1990s, touch DNA concept came into the picture. First ever documented case of touch DNA was found in 1997, where DNA were extracted from epithelial cells by Van Alscott and Mr. Jones. There are several techniques of collecting DNA. First one is swabbing technique. Dry sterile swabs are used to collect DNA. Traditionally, collect DNA from the object. This method was used traditionally. Later on, wet swabbing technique came into the picture. So the cotton swabs were made wet with the help of sterile liquid or non-porous surfactant. This method worked well because non-porous surfactant reacted well with cationic DNA. This helped retrieving more DNA cells. Second method is tape lifting. Tape lifting method is used especially on fabric objects because their swabbing technique may cause contamination. So water soluble tapes are used to collect the DNA samples. Third method is cutting or scraping of the object. So actual physical evidence or actual object is cut or scrapped off, and then DNA samples are get collected. But this method destroys actual evidence at the crime scene. Last method is DNA collection with the help of vacuum, where the sterile solution is spread over the object and then the liquid containing DNA sample is collected with the help of vacuum. The mechanism of touch DNA is based on Locard's principle, where it states that when two objects come into contact, they exchange traces with each other. The process of touch DNA includes several steps such as cell lysis, then amplification, cell electrophoresis, and creating profile of STP, creating DNA profiles. So when person touches to an object, their screen cell gets shed off on that object. With the help of mentioned methods of collecting DNA, one of the method is used to collect DNA sample. As this sample is very minute, this sample is further sent to amplification. With help of polymerase chain reaction, the sample is amplified. This amplified sample is further sent to electrophoresis to form DNA fragments. These DNA fragments further with the help of next generation sequences, analyzed with the help of next generation sequences. This method is used to create to get knowledge about the libraries of these genes. The incorporation of touch DNA in criminal investigation has been more impactful, as it helps to resolve cold cases which remain unsolved in the past, or it can reduce wrong convictions. As every coin has two sides, touch DNA also has some limitations. There are chances of contamination due to several factors. It might happen that the investigator's DNA might get collected because of a wrong method of collecting DNA. There are some future aspects to this touch DNA, where a portable DNA technique can be incorporated to collect DNA from actual criminal site and analyzed over there itself, so that it can reduce the chances of contamination. Thank you.